Good day! Today we will be discussing about prokaryotic organisms. Our lecture slides comes from Visualizing Microbiology, A Healthy Perspective by Rodney Anderson and Linda Young. So when we talk about prokaryotic organisms, we are talking about the organisms in the microbial world that do not have a nucleus. So when we look at them in a microscopically, so we can actually see them like so. So here is a micrograph of a colored micrograph of a prokaryotic organism. These are the yellow blobs on a fiber. So they are living atop a fiber. Or rather a fibrous material. And then here we have your agar plate. So you have a petri dish containing a culture of your prokaryotic organisms. So first of all, let's define prokaryotic organisms or prokaryotes. So they are small unicellular organisms. So that means their whole body is just made up of one cell. So they lack nucleus and membrane-bound organelles. And this is actually one of the main differences they have over the other group of cells, which are the eukaryotic organisms or the eukaryotes. So under the prokaryotes, we have the bacteria and the archaea. They are, as I said before, they are distinct from eukaryotic organisms. So some prokaryotes can be pathogenic, but some of them can also be beneficial to us. So here is a comparison, a table of comparing the prokaryotes and the eukaryotes. So in terms of size, we have prokaryotes that are much smaller, significantly smaller than the eukaryotes. So as you can see, they range from around 0.2 to 5 micrometers, whereas the eukaryotes, the sizes of the eukaryotes are around 10 to 100 micrometers. This is because eukaryotes have nucleus, they have membrane-bound organelles inside them, so which makes them bigger. As for the genetic material, the genetic information inside the eukaryotes is made up of a circular DNA, that's their chromosomal DNA, and then we also have smaller independent circular DNA molecules separate from the chromosomal DNA. We call them the plasmids. They carry the accessory genes. And then for the eukaryotes, the genetic information are encoded into several linear chromosomes. These are the iconic X that we see when we talk about genes. So this actually comes from the the image of the genetic material inside the eukaryotes. Now, for the protein synthesis, prokaryotes synthesize proteins using ribosomes. The same way with the eukaryotes. The eukaryotes also synthesize proteins using ribosomes. But the main difference is that the ribosomes of the prokaryotes are smaller, they are less dense. Hence, it's called the 70S ribosomes. It indicates the S value, which indicates their density. So, it's a 70S. So, smaller compared to the eukaryotic value, which is 80 or 80S. And then for the cell wall of the prokaryotes are more structurally complex and made up of peptidoglycan layers versus those of the eukaryotes, which some eukaryotes even do not have a cell wall, such as for us humans. We are made up of eukaryotic cells, and we do not have a cell wall. And for those that have a cell wall, such as the plants, they are usually structurally simple. One of the main reason is because for the prokaryotes, that one cell is all they have. So when that cell that they have dies because they do not have proper protection of the cell wall, of course they will die. Well, whereas for us, if some of our cells die, it's okay. So we also have other cells that can continue on living for us. So basically, that's one of the main reasons. Now, we have, aside from that, we also have the DNA organization. So in terms of the organization of the DNA, the DNA in prokaryotes, since they do not have a nucleus, so they are no membrane-bound nucleus. So the DNA is just just in a region that has really no boundaries inside the cell. And then we have the eukaryotes, whereas there is a membrane-bound nucleus here. So that means the DNA is separated from the prokaryotes, or rather, separated from the rest of the cytoplasm. So they are much more protected. The DNA of the eukaryotes are protected. And then we have the organelles. There is no true membrane-bound organelles for the prokaryotes, although they have inclusion bodies. But for the eukaryotes, they have many complex membrane-bound organelles. For the cell division, they replicate by binary fission. That's for the prokaryotes. or which is a type of a cell division, a simple cell division process that separates the copied nucleoids into the daughter cells. Whereas for the eukaryotes, there are two ways. You have the mitosis and the meiosis for the sexual reproduction or sexual division. So, basically, if you remember your general biology back in high school, so it's basically the mitosis. We have several stages of mitosis where chromosomes line up. So that is applicable for eukaryotes, not necessarily for prokaryotes. Now, an example of an organism, we have the E. coli for the prokaryotes and the Euglena gracilis here for eukaryotes. Euglena is a single-celled eukaryotic organism. So, the whole organism is just made up of one cell itself. So, that's the Euglena. Okay, so the prokaryotes, actually the bacteria, are rather the microbial organisms. They are the dominant form of life on Earth. So the prokaryotes are widely distributed and live in almost every possible environment. Because part of the prokaryotes, or under the prokaryotes, we have the Archaea, which are the special, these are the organisms that can and does live in extreme environments. We have the extremophiles. We usually call them extremophiles. Extreme hot, extreme cold, extreme acidity, extreme salinity, extreme alkalinity. So basically, whenever you have your extreme environment, that's where they are found. If you culture them away from that environment, they usually don't live, they die. So total prokaryotic biomass is 10,000 times greater than the total biomass of the global human population. So we have 10 times more bacteria living in and on our bodies than there are human cells, because human cells, or rather the bacterial cells, are much smaller. So here is a rough estimate of the amount of bacteria that we have. So we have bacteria present in the water vapor, in the clouds. So we have those on the surface of the leaves of the plants. We have them in our mouth, on our skin, in our intestines, on the surface of the soil, even in the rumen of the grazing animals, in the ponds, rivers, and waters. So here is the estimated number of species. These are estimates because especially for the case of bacteria and archaea, we still do not know the exact value. So these are just the rough estimates. So we have here a billion for the bacteria, and for the archaea it's to be determined, because actually they are really very difficult to characterize and culture, because again, they require extreme conditions for us to culture them, and it's very difficult to replicate those extreme conditions in the lab. So, prokaryotes help sustain the life on Earth. So, for example, we have the photosynthetic organisms, prokaryotic organisms that produce the oxygen and glucose. They also perform nitrogen fixation. So, basically, for nitrogen fixation, they get the inert nitrogen gas in the atmosphere and makes them into usable nitrogen that we can use to actually create amino acids and other biomolecules. So, they help ruminants break down cellulose as well. It's important to break down cellulose because, for example, the plant matter, we cannot decompose plant matter in our stomachs. But for the animals that graze or that actually subsist on vegetables, the herbivores, they must be able to actually break down the cellulose for them to get the energy from the cellulose. Since it's their main form of or main source of diet, so the bacteria help them do that. And then we also have involvement of the prokaryotes in the biogeochemical cycles. So we have important components in the food cycle, especially for the bacteria that are in the decomposers. So they help basically they help complete the cycle, the food cycle. So in terms of its interaction with other organisms, we have symbiotic relationships, whereas one organism lives in or on another, which is actually the norm for prokaryotes because they are very, very small, and then they live on the surface or inside other organisms. So prokaryotes often form symbiotic relationships with the eukaryotes. So they are classified based on how the other species is affected by this interaction. So we have the mutualism, commensalism, and parasitism type of interaction. So here you have the range of the range of relationships that we can have. So we have the mutualism, whereas there's a mutual benefit. For example, the bacteria living in the root nodules of plants, they help in the nitrogen fixation, and in doing so, helps the plants in actually acquiring fixed nitrogen, so nitrogen that can be used to produce biomolecules. And then, so basically, they benefit themselves and benefit their host. So for the neutral interactions, you have your Enterococcus fecalis. They grow inside our intestine, and there's not no yet known functions of those microorganisms that benefits us, but it benefits them because we provide them a home, and of course, food. And then we have the parasitism. So basically, this is a harmful interaction with the host. So we have the Argobacterium tumefacens. It's a type of microorganisms that causes the crown gall diseases in plants. So we have since the bacteria, they are quite diverse. They are actually quite difficult to identify, or rather, it's quite a challenge to identify them absolutely. So one of the ways to identify them or classify them is through their shapes. It's the most common or the easiest because one of the easiest because we can just use a microscope to view them. So their size ranges from 0.2 to 2.0 micrometers in diameter and are 2 to 8 micrometers in length. So their cell wall structure actually determines shape. So it actually helps us to classify them into their taxonomic classifications. So, and actually, if you look at the names of the bacteria, most of them pertain to their shapes. So, three of the most common shapes we have the coccus and the or cocci, we have the bacillus or the plural bacilli, and then we have the spiral shapes. So we also have pleomorphic cells. So these are cells that have irregular shapes. So here are some example, or rather the meanings of behind those names in the bacterial shapes. So when we talk about bacillus, we are talking about the rods or the stuff. So they're elongated, somewhat like hot dogs. So elongated bacteria. So we have the Vibrio or wave-like. Basically, they are the curved ones. Spirillum, they are the coiled. The coccus or kernel, they are the circular bacteria. And then the spirochete. So similar to a spirillum, but much kinkier. So they have, it's actually, the texture is similar to a coiled, very, very coiled curly hair. So, and then other terms based on the groupings when we look at them in groups, multiple bacteria in groups. So you have here diplo, which means double. So that means two bacterium connected to one another. So we have strepto, they form a twisted chain. Staphylo, they form a cluster. The tetra, they form groups of four, or a sarcinae, which forms a three-dimensional bundle. Actually, to better visualize them, here we have the different shapes of the bacteria. So for the cocci or the coccus, they are actually circular. We have the bacilli, which are rod-shaped. And then we also have a subgroup for the bacillus, the bacilli, which are actually, if you notice, it's not two cocci, but rather they are fused. They are fused to one another, so they share the same cytoplasm, but they have rather indentations in the middle. So they are like bacilli, but with indentations in the middle that makes it appear like we have two circular bacteria. Then we have the Vibrio, the curved ones, the spirilla, spiral, and then the spirochetes. A more really, not they are actually more straight, but if you look at them, they have tight waves. And then you have the unusual one, the fusiforms, and the club-shaped rods. And then when we look at them with pairs, so we have the diplococci and diplobacilli. And then we have the streptococci and the streptobacilli. For the three-dimensional packets, you have the staphylococci that they look like a bunch of grapes. You have the tetrads and the sarcinae. So sarcinae and tetrads, it's quite similar, but for the sarcinae, it's a three-dimensional one. So it's a 2 by 2 by 2 dimensions of your coccus bacterium. Whereas the tetrads is just a two-dimensional for arrangement of 4. So 2 by 2 arrangement. So you have, aside from that, we have filamentous bacteria for the more unusual ones, the hyphal. Bacteria that forms a hyphal morphology. So they form similar to roots. And then you have appendage bacteria. So these are some of the shapes of the bacterium. So we have, in terms of bacterial arrangement, we have species-specific groupings of the cells. So the cell shape and arrangement can be used to diagnose them. So, for example, for that is the Seria gonorrhoeae. So, some common arrangement that we have in pairs, chains, clusters, tetrads, and palisades. So, this is an example of a palisade arrangement. So that's it for now for this part of the lecture. So in the next part, we will be discussing more in depth about the different parts of your bacteria.